kaki dia ACL dengan meniscus. Tak, dia cakap lutut dia meletup. Sekejap, saya mute. Alright guys, hari tu kita stop kat mana Ke hari tu lah, hari Isnin. Kita stop kat mana Isnin tu? Assalamualaikum.
Salah, dekat legislating.
DNA replication dah habis belum? DNA replication dah habis belum? Habis belum?
Dah medium.
Dah, okey. Kalau dah, kenapa ada yang tak jawab lagi Saya punya app puzzle ni ada lagi 9 orang. 9 pula. Ada berapa orang? 8. Ada lagi berapa orang yang tak jawab? So tolong jawab lah. Okay, saya tak nak sebut nama, you know who. Okay, yang lain yang dah jawab, okay bagus. Yang dah jawab tu memang jawab lah. Yang tengok video tu memang tengok and jawab. Sebab mostly yang dapat 86 sampai 100 tu memang betul lah dia tengok. Bukan main jawab sahaja. So tolonglah awak patut bersyukur sebab sebab topik ni masuk sebagai assignment, bukannya sebagai awak punya exam. So exam memang tak tahu lah nak cakap macam mana. Okay, so DNA replication kita dah habis discuss. So before we start, okay, what is the model of DNA replication, Alia Shakira? What is the model of DNA replication, Alia Shakira? Alia Shakira. Alia Shakira.
Yes, madam.
Yeah.
Semi-conservative.
Semi-conservative. Good. Okay, what are the thank you Alia. What are the other two models? Alia Najwa. The other two models of DNA replication?
Conservative dengan dispersive.
Conservative and dispersive, very good. Okay. DNA replication, we have 6 enzymes and 1 protein. Sarah Sofia, bagi saya satu enzim.
DNA helicase.
DNA helicase, very good. Hana, bagi satu.
Primase.
Primase, Sakinah, satu.
Helicase. DNA helicase.
Helicase ganda ke?
Ligase.
Ligase, okay. DNA ligase, very good. Syafri, bagi satu. Syafri.
Topoisomerase.
Topoisomerase, very good. Amirah Alia, satu. Amirah Alia. Amirah Alia Hanim. DNA polymerase 1, very good, thank you. Anas, satu lagi.
DNA polymerase 3.
DNA polymerase 3, okay, bagus. Alright, so 6 enzymes and 1 protein. Alright, for today we're going to discuss about transcription. 2 jam kelas kita, ya Allah. Transcription. Okay, kita akan discuss pasal transcription, protein synthesis, transcription and translation. Okay, so I'm going to share my screen with you. Sebentar. Okay. Okay. Right, so protein synthesis, transcription, translation. Okay, protein synthesis, transcription and translation. For protein synthesis, we have 2 steps. We have 2 processes, transcription and translation. Kenapa dia tak keluar? Okay. Okay, transcription, translation. So for today, we're going to discuss 6.3. Okay, we're going to discuss 6.3, transcription, translation. And these 2 processes occur in nucleus and cytoplasm. So, but before we proceed to transcription and translation, protein synthesis, okay, protein synthesis takes ribosome. Okay, ribosome is the one is a function of ribosome is to synthesize protein. Okay, and the ribosome can be found or attached on ER, or it can be found freely in cytoplasm. Okay, and transcription is the process where we're going to produce mRNA from DNA, and translation, we're going to produce protein from RNA. Yep. Okay, so these are the learning objective for today. Explain briefly transcription and translation. Introduce codon and its relationship with sequence of amino acid using genetic code table. Introduce codon. Codon kita ada start codon and stop codon. Siapa tahu apa kita punya start codon? Start codon. Apa kita punya start codon? AUG. Okay, apa pak kita? Thank you Diana. Apa kita punya stop codon?
UAA.
Lagi? Ada 3.
UGA.
Lagi?
UAG.
Okay, kita punya stop codon ada 3, uwa, uga. Okay, uwa, uga adalah kita punya stop codon. C, explain transcription and the stages involved. Okay, we have transcription, we have 3 stages: initiation, elongation, and termination. And in the formation of mRNA strand from 5' to 3'. Explain translation and the stages involved in translation. Sama juga, initiation, elongation, termination. But for elongation, we have 3 sub-stages: codon recognition, peptide bond formation, and translocation. Okay, so before we proceed on transcription, I'm going to give overview on transcription and translation, and I'm going to introduce codon and its relationship with sequence of amino acids using genetic code table. Okay, so protein synthesis, we have transcription. As I said, it occurs in nucleus, okay, from DNA to mRNA, and we have 3 sub-stages: initiation, elongation, and termination. After mRNA is synthesized, mRNA will exit the nucleus. mRNA ni keluar daripada nucleus. Macam mana dia boleh keluar? Siapa tahu macam mana dia boleh keluar? Dia keluar. Kat mana dia keluar?
Ada orang ER.
Kat mana?
Dalam ER.
Dalam ER.
Is it at the nucleus pores?
Yes, Jude. Thank you. Okay, thank you for trying, Ketina. Okay, macam Jude cakap, it exit through nuclear pore. Okay, nuclear pore. Ingat lagi tak structure nucleus? Ingat lagi tak?
Yeah.
Yeah. Okay, iya. Okay, structure of nucleus kita ada nuclear pore. Kita kita ada nucleolus, kita ada nucleoplasm. Okay, so this mRNA will exit from nucleus through nuclear pore to the cytoplasm. So once it reach cytoplasm, there is process called translation. We going to translate. Okay, kita akan translate. Okay, contoh mRNA ni, we going to translate an mRNA into polypeptide chain, into protein. Okay, macam kita buat, biasa tak guna Google Translate? Biasa kan? Kalau Google Translate, selalunya kalau kita Google Translate yang bahasa yang kita tak tahu. Contoh saya nak Google Translate bahasa Espana, okay, bahasa Spanish, saya nak translate. So daripada Spanish kepada bahasa yang saya faham, English or Malay. So nak translate tu, kita guna Google Translate lah. Okay, kita nak Google Translate lah. Sama juga translation yang ni. Kita nak translate mRNA, nak translate genetic code dalam mRNA kepada satu benda yang kita boleh baca, iaitu polypeptide chain or protein. Still remember protein? Ingat tak lagi protein? Ingat tak lagi chapter 1 protein, topik 1 protein, apa dia? Ingat tak apa monomer of protein?
Amino acid.
Amino acid, what are the structures of amino acid? What is the basic structure of amino acid? Krik-krik. Krik-krik yang malas ni. What is the basic structure of amino acid, guys?
Amino group.
Amino group lagi.
Carboxyl group.
Apa dia?
Carboxyl group.
Okay, carboxyl group. Okay, siapa yang
R group.
amino acid group, carboxyl group, R group lagi? Apa? Satu lagi?
R group.
R group dah. Satu lagi.
Hydrogen.
Hydrogen atom. R group ada 4. Apa dia?
Polar.
Acid.
Acidic, basic,
Non-polar.
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 Bagus. Okay, ya, pandai. Polar, non-polar, basic, acidic. Okay, bagus. Maksudnya awak memang study lah. Bagus, korang study. Saya suka. Okay. For translation, we also have three stages: initiation, elongation, and termination. As I said, elongation, we have 3 substage: codon recognition, peptide bond formation, translocation. For protein synthesis, okay, for each stage in protein synthesis, for each process in protein synthesis, for transcription, we need 3 things. Okay, we need 3 items in order for transcription process to occur. First, we need promoter. Then we need RNA polymerase, the enzyme RNA polymerase. And lastly, we need terminator. DNA mRNA tu memanglah ada. Okay, tapi 3 benda ni kena ada. Promoter, RNA polymerase and terminator. For translation, we need ribosomes, tRNA, amino acids. Okay, so first I'm going to explain about transcription. But before that, I'm going to explain about genetic code and its relationship with sequence of amino acids. So as you already know, DNA is double-stranded. Okay, DNA is double-stranded. What is the other characteristic of DNA? Apa lagi characteristic of DNA that you need to know?
Anti-parallel.
Okay, DNA is anti-parallel. Dia run in opposite direction. For example, 5' to 3', 3' to 5'. Non-template strand and DNA template strand. From DNA to mRNA, we have a process called transcription. Okay, the product of transcription is mRNA, which run from 5' to 3'. Along the mRNA, we have codons. Okay, codon is read in triplet base. Kita baca codon dalam bentuk triplet. Triplet. Tahu tak triplet apa? Kembar 3. Tahu tak triplet?
Tahu.
Triplet, okay? Song triplet bases. So kita okay, codon. Codon. Kita baca dalam triplet. Codon. Dekat mRNA, along mRNA, we have codon. And what you need to know about codon, we have start codon. Start codon is AUG. Stop codon, we have 3. Uwa, uwa, uga. Okay, uwa, uwa, uga. In this diagram, we have stop codon UA, UAA. Okay, so that is one part. From mRNA to polypeptide chain, we have a process called translation. Okay, from mRNA to polypeptide chain, we have a process called translation. Okay, translation. We're going to translate these codons into polypeptide chain or protein. How we going to translate? We're going to translate all this codon by referring to genetic code table. By referring to genetic code table. You don't have to remember. You don't have to remember the genetic code table. Tak perlu, nanti dia akan bagi. Dia akan beri awak genetic code table ni. Okay? So, for example, if we have AUG, the start codon, the amino acid is methionine. Okay, methionine. So, kita tengok dekat genetic code table, AUG. So, the amino acid is methionine. Right? Then next we move on to the next codon, which is AGC. So, we refer to the genetic code table, AGC. What is AGC? So the amino acid for AGC is serine. Serine. So serine lah dia punya nama amino acid kat sini. Sampai until it reach stop codons. Once it reach stop codon, for example, we have UA, UAA. UAA, stop. UAA does not mean anything. Okay, UAA tak membawa maksud any amino acids. So that's why dekat sini kita tak ada amino acids. Right? Okay, itu adalah proses translation. That is overview. What is translation all about? For transcription, okay, for DNA to mRNA, mRNA is single-stranded, which run from 5' to 3'. So when we going to transcribe, okay, kita nak transcribe, we going to transcribe which strand, which of these 2 strands of DNA that we going to transcribe. Yang mana satu dalam bahasa Melayu ni, bahasa senang awak faham. Okay. Antara 5' ke 3' dengan 3' kepada 5', yang mana satu kita nak transcribe menjadi mRNA. Which strand?
3'.
3' to 5'.
Yang lain? Yang lain?
3', 5'.
Kenapa? Kenapa?
Sebab dia yang baru.
Ha?
Lain.
Kenapa? Kenapa?
Kan kalau DNA replication kan, 3' ke 5' tu macam yang baru punya. Tak ada.
Tak. Dia tak ada kaitan dengan DNA replication. Kenapa? But thank you for trying, Sakinah. Okay, you are very brave compared to others yang senyap ni. Tidur ke? Orang tidur ke?
Madam,
Ha.
not, I think it's 5' to 3' because the mRNA is 5' to 3' also.
Okay, so sama lah. Okay, good try. Thank you, Jude. For
Madam?
Yeah.
It can be 5' to 3', right? Because the DNA primase only works from 5 to 3.
Okay. 5 to 3. Alright. Okay. Yang lain. Oh, menarik lah perbincangan ni. Yang lain, boleh tak? Yang lain?
Madam, I think it's 5 prime to 3 prime.
5 prime to 3 prime also. Okay, so we have two answer. We have two options lah. Okay, we have two options, 5 prime to 3 prime. Siapa yang agree 5 prime to 3 prime, angkat tangan. Okay, Adab, bagi reason. Adam, Safwan, bagi reason. What is your reason?
Sebab dia anti-parallel.
Sebab dia anti-parallel. Okay, thank you. Bukan betul tapi thank you bagi reason. Kalau bagi reason tak tahu tu saya tak suka. Aiman, why? Muhammad Aiman. Muhammad Aiman. Kita banyak masa, 2 jam kot. Memang saya boleh relax-relax, tanya, ambil masa. Tak dengar kau, tak dengar hang. Kalau saya dengar, saya bercakap lah. Tak dengar apa. Tapi saya dengar notification masuk. Sebab dia non-template. Sebab dia non-template. Okay. Siapa yang cakap 3 prime to 5 prime? Angkat tangan. Izati, Atira, awak cakap 2 kali. Tadi awak angkat tangan, ni awak angkat tangan.
Saya lupa turunkan, madam.
Okay. Anas, kenapa?
Sebab kan transcription kena ada RNA polymerase kan. RNA polymerase tu dia cuma buat daripada 5 prime ke 3 prime kan.
Okay.
Dia akan ikut template lah. Dia akan ikut 3 prime ke 5 prime. Sebabkan anti parallel dia buat 5 ke 3. Jadi
Ya, itulah jawapan saya cari, Anas. Thank you. Ya, dia akan ambil yang ini. Okay guys, macam Anas cakap tadi, dia akan copy yang ini. 3 prime, 5 prime. Dia akan copy DNA template strand. First reason kenapa, sebab mRNA adalah 5 prime, 3 prime. Okay, ada 2 reason lah. Antara reason dia adalah mRNA adalah 5 prime kepada 3 prime. So dia kena copy yang 3 prime. Yang kedua, Anas bagi tahu jawapan dia tadi. Sebab RNA polymerase hanya boleh bergerak daripada 5 prime kepada 3 prime. Okay? Itu jawapan dia. So bila kita nak cari, bila kita nak transcribe, contoh soalan dah bagilah. Soalan dah bagi ni. Okay, soalan dah bagi ni, kita nak transcribe. Okay, soalan dah bagi, kita nak transcribe. Contoh dia katalah dia tak label kan. Bila kita nak transcribe, awak kena transcribe, awak kena tengok yang 3 prime yang ni. Tengok yang ni. Inilah yang transcribe yang ni, transcription yang ni lah awak akan guna. Strand yang ni lah. Okay, strand 3 prime kepada 5 prime ni lah yang awak akan transcribe menjadi mRNA. So for example, T complementary with A, G complementary with C, A complementary with U, C, G, GC, GC, CC. Sampailah habis. I repeat, apa yang you nak copy, you kena copy yang 3 prime kepada 5 prime. Okay? Right. Boleh ke?
Boleh.
Okay, jelas jelas dan nyata. Okay. So ada yang apa namanya, so bukanlah kita berapa belas orang tadi, berapa 15 orang kot yang angkat 5 prime 3 prime tu tadi, itu adalah reason dia kenapa kita pilih 3 prime kepada 5 prime. Okay, kita dah cerita pasal codon, kita ada juga anti-codon. Okay, fan dengan anti-fan. Codon, anti-codon. Codon duduk dekat mRNA, anti-codon dia duduk dekat tRNA. Okay, for example, tRNA, ada tak gambar tRNA? This one. This is tRNA. Okay, this is a tRNA. tRNA is fold into 3D shape, and this site is amino acid attachment site. And this region is where anticodon is located. Okay, anticodon located dekat bawah kepala dia, ada amino acid attachment. So basically kalau kita lukis tRNA, ni tRNA, yang ni. Ni adalah tRNA. So kalau tRNA, ni adalah tRNA. tRNA kepala dia ada amino acid, kaki dia ada anticodon. Itu adalah tRNA. T tu maksud dia apa? Apa maksud T? Transfer RNA. Okay, tRNA. Alright? Okay, untuk genetic code table okay ke? Okay ke untuk genetic code table?
Okay.
Alright, so we're going to discuss transcription. So as I said, transcription occur at the, where does transcription occur?
Nucleus.
That's good. Okay, what is the items, what are the things that we need in order for transcription to occur?
Promoter.
What else?
RNA polymerase.
RNA polymerase. Lagi?
Terminator.
Korang pernah tengok tak cerita Terminator tu?
Pernah.
Kan? Terminator, I'll be back. Pernah tengok? Oh, bagus. Kan? I'll be back. Okay, yang itulah. So, kita tengok dulu gambar ni. So, this is promoter. Okay, this is promoter. This is RNA polymerase. Okay, RNA polymerase. Start point, this start points maksudnya this is where transcription start. Okay, transcription occur at this region. Transcription is occur at the promoter region. Transcription unit refer to the region where it will be transcribed into RNA transcript. Okay, so in transcription, we only going to transcribe one strand. Okay, we only going to transcribe one strand. Okay, which strand kita akan transcribe? Yang mana satu kita akan transcribe? Which strand?
3, 5.
3 prime to 5 prime. Okay, 3 prime to 5 prime. So we're going to transcribe from 3 prime to 5 prime. Alright, we only going to transcribe one strand only. Unlike DNA replication, we're going to transcribe both strand. Transcription, satu strand saja. Actually, ini adalah salah satu dululah, bukan tak adalah dulu sangat, 3-4 tahun lepas, soalan selalu tanya differences between transcription and DNA replication. Okay, soalan favourite tu, sangat favourite. Okay, tapi tak adalah, untuk kali ni saya bagi tahu sikit je. Okay, so what happened here, this RNA polymerase, it will bind. Okay, RNA polymerase. Sebentar. Ni ke? Saya salah ni. Sekejap, sebentar. Okay. RNA polymerase. Okay, keyword dekat mas sini. Pen lah. Okay. Okay, here, this is when RNA polymerase will bind. Kenapa color merah? You kena tukar ke? Belum tukar lagi. Okay, this is when RNA polymerase, it will bind to the, okay, RNA polymerase will bind to the promoter. So when it bind to the promoter, this DNA will unwind the double helix. Okay, it will unwind the double helix. Okay, it will unwind the double helix. Wait. Kejap Apa benda lah ni? Okay, payah okay. DNA akan unwind the double helix. So itu adalah proses initiation. Okay, this is a process of initiation. Okay, initiation. Yang ni adalah elongation. Elongation maksudnya dia akan bergerak. Okay, RNA polymerase ni akan bergerak, move along the DNA. Okay, bila dia bergerak tu, dia bukan bergerak saja, it will catalyze the addition of RNA nucleotide until it reach terminator. So once it reach terminator, the transcription will be stopped and mRNA will be released. Okay, so that is overview. So kita tengok the first stage. First stage is initiation. Okay, initiation. Initiation maksudnya apa? Apa maksud initiation, guys? Ha? Apa dia? Permulaan berwawasan. Okay, permulaan berwawasan. Lagu apa Oh tak, itu lagu ni. Lagu Perak nombor 1. Okay, so initiation adalah permulaan. Bila permulaan, apa yang kita kena buat adalah, apa yang awak kena cerita adalah RNA polymerase. RNA polymerase ni sekarang ni masa permulaan, masa initiation, dia buat apa? It will bind. Okay, it will bind. Tak lawa. Padam balik. Okay, it will bind. Okay, it will bind. Sini. Okay, it will bind. Bila it will bind, dia akan bind dengan siapa? It will bind to the promoter region. Siapa promoter region? Awak tak perlu tahu. Okay, awak tak perlu tahu. After it bind to the promoter region, it will unwind the double helix. Dia bind, barulah dia unwind. Maksudnya bila dia unwind tu, maksudnya dia akan buka double helix. So bila dia buka double helix, dia akan jadi bentuk macam ni tau. Dia akan jadi bentuk macam ni. Okay, yang ni, macam ni. Yang ni kita panggil sebagai replication bubble. Okay, replication bubble. Right? And only one strand of the DNA act as template. Okay, that is initiation. After initiation, we have process called elongation. Okay, proses elongation, proses bukan memanjang. Dia proses, bukan memanjang. Dia penambahan lah. Okay, sebab dia nak hasilkan mRNA kan. So elongation ni, this is when the RNA polymerase move along. Okay, sekarang RNA polymerase tengah buat apa? RNA polymerase will move along the template. Yang ni ribosom sebesar alam ni, ribosom, RNA polymerase sebesar alam tu, itu adalah enzim. It will move along the template. So once it move along the template, okay, dia bergerak. This is ni, dia akan bergerak. Move along the template. Move je. Move. Masa gerak-gerak tu, it will catalyze the addition of RNA nucleotide. Maksudnya masa tengah dia bergerak, it will catalyze the addition of RNA nucleotide. Dia akan tambah RNA nucleotide to the 3' end of the growing strand. And elongation of RNA transcript is in 5' to 3' direction. Okay, so when it catalyzes the addition, for example, if the DNA is TGA, okay, if the DNA is TGA, then RNA nucleotide will be complementary, ACU. Okay, dia akan tambah macam tu. Right? And then the elongation process will be continue until it reach terminator. I'll be back. Okay. Until it reach terminator. Okay, once it reach terminator, RNA polymerase will stop transcribe. Stop transcribe. Okay, dah habis dah kerja dia. Dia stop. Once it stop, mRNA will be released. Okay, mRNA will be released and the RNA polymerase will be detached from the DNA, and DNA daripada macam ni, DNA daripada macam ni, it will back to the double helix. Okay, dia akan balik-balik ke dalam bentuk double helix. After mRNA is released, mRNA nak pergi mana? mRNA nak pergi mana?
Cytoplasm.
Kenapa nak pergi cytoplasm, Sakina?
Dia nak lakukan translation.
Sebab dia nak buat proses translation. Okay, dia nak buat proses translation. Alright, setakat ni okey ke? Boleh ke transcription?
Okay.
Okay.
Amir ada kena Amir. Anas okey?
Okay.
Aiman okey? Adam? Aiman?
Ya.
Okey ke?
Okey.
Okay. Okay guys. Ada dengar berita ke nak masuk college ni?
Ada.
Ada ke? Bila?
Ada.
17
Tapi saya dah
Bulan 10? Bulan 10 tu berapa hari bulan?
kot.
Apa?
17. Tak tahu.
13?
ada cakap
17 kot tak? Katanya.
Tapi bulan 10.
Bulan itu lah.
Betul ke Megan?
Cara yang benar dua-dua macam mana?
Tak payah balik rumah.
17
Vaksin.
Sunday.
Kena vaksin.
Tak tahu nak cakap macam mana.
Tapi Puan Latifah kata tak betul. Ada yang dah tanya Puan Latifah kata tak.
Ikut Puan Siti Latifah cakap lah. Okay, dia lebih tahu. Sebab dia apa pun info akan dia dapat dulu. And kita still fasa 2. Okay, kita still fasa 2. Fasa 2 still online. Tapi kita tak tahulah. Tak tahu. Untuk masa terdekat ni tak. Tapi kalau tiba-tiba kita masuk fasa 3, awak get ready lah untuk yang siapa yang dah second dose. Siapa yang dah second dose angkat tangan. Siapa dah complete vaccination? Oh ramai. Berapa orang? 13 orang. Oh lagi 16. Oh ramai juga tak second dose lagi. Okay. Apalah. Penat online ni. Okay. Penat. Alright. So kita doa je lah mana yang terbaik. Okay. Ataupun online ataupun face to face. Okay, kita doa je lah mana yang terbaik untuk kita semua. Alright. Okay, sambung untuk ni. Okay, sambung untuk transcription. Okay, transcription dah lepas dapat mRNA. Okay, lepas dapat mRNA tu, in eukaryotic cell, okay, for example, what is the example of eukaryotic cell? What is the example of eukaryotic cell?
Plant cell.
Animal cell.
Plant cell, animal cell. Okay, plant cell, animal cell. So in eukaryotic cell, mRNA is modified after transcription. Okay, mRNA tu, it will be modified. It will undergo process called RNA splicing. Okay, process called RNA splicing. Okay, during RNA splicing, this is when the intron, okay, the non-coding region, the intron will be removed. In our DNA, okay, DNA of eukaryotic cell, we have exon region and intron region. Okay, during transcription, exon and intron region will be transcribed. And then we have a product, RNA, which contain exon and intron. But we only want exon. Kita nak exon saja. Okay, kita nak exon saja. Kita nak exon saja, bukan exo Kita nak exon saja. Kita nak exon sebab apa? Kita nak exon sebab dia adalah coding region. Okay, dia adalah coding region. Maksudnya dia akan membawa maksud. Kod dia tu ada maksud. Intron is the non-coding region. So we don't want intron. That's why after mRNA is produced, RNA will undergo RNA splicing. Okay, so what happen is this intron will be removed. Okay, this intron will be removed. It will be removed by a protein called spliceosome. Okay, intron will be removed by a protein called spliceosome. After intron is removed, this exon will splice together. Okay, it will splice together. Dia akan dicantumkan. And we get mRNA which only has exons. Okay, now we have mRNA that only has exon. Exon yang beri maksud. Exon yang which is coding region, coding sequence. After that, mRNA will exit the nucleus through nuclear pore to go to cytoplasm. Okay, in prokaryotic cell, in prokaryote, what is the example of prokaryote?
Bacteria.
Bacteria.
Yeah, bacteria is example of prokaryotic cell. Okay, so bacteria do not has intron. Bacteria memang ada exon saja, so dia tak perlu undergo RNA splicing. Only eukaryotic cell will undergo RNA splicing. Okay, boleh ke?
Boleh.
Boleh.
Okay, boleh. Perlu break 5 minutes ke? Ke nak continue? Apa yang angkat tangan? Jude, ya, kenapa? Kenapa angkat tangan? Angkat tangan untuk continue ke angkat tangan untuk break?
No, I need to go to toilet.
Yeah, please have a break for 5 minutes. Okay, kita break dulu 5 minutes. Ada apa-apa nak tanya, boleh tanya sekarang. Okay, 2.50 kita sambung. Ada soalan nak tanya, boleh tanya sekarang lah. Kita break dulu kejap. Okay.
Madam,
Hmm.
can you explain like the part where after the mRNA is produced, it goes to the cytoplasm part? That part, can you explain again? Because my line was lagging just now, so I couldn't hear.
Alright. So after mRNA, after we have our raw RNA, this is our raw RNA, this one. Okay, this is our raw RNA. Okay, this raw RNA will undergo process called RNA splicing. Why? Because we want to remove the intron. Because intron is non-coding region. Right? So after we remove the intron, the non-coding region, the exon will splice together. Okay, we're going to cantumkan the exon, and then we get the mRNA which only has coding sequence. Okay, maksudnya coding sequence ni membawa dia adalah sequence yang membawa maksud. Maksudnya bila kita translate, dia membawa maksud satu amino acid. Kiranya bila kita translate tu, oh, dia ada amino acid dia. Alright? So that is mRNA. mRNA yang ada coding sequence. Dapat je mRNA yang cantik macam ni, yang tak ada intron, yang hanya ada exon, this mRNA will exit the nucleus. It will go to the cytoplasm. Okay, it will exit through nuclear pore. Nuclear pore, dia akan keluar ni lah. Okay, at the cytoplasm, then we have process called translation. Okay, Katina?
Oh yeah, okay. Yeah, I got it. Okay, thank you, madam.
Madam.
Ya.
Maksudnya RNA splicing tu the function dia is to remove the introns lah.
Ya, betul. Pandai.
Thank you, madam.
Madam, I have another question.
Yeah.
Regarding the genetic food table, right?
Yeah.
The codon, it doesn't bring any meaning, like what you said earlier. So, does that mean that any of the amino acid can become a start codon also?
Start codon only AUG. The start codon only AUG.
Or only AUG? So
Yes.
the threonine cannot become, or the
Cannot.
protein cannot. Oh, okay.
Cannot. Only AUG. Only methionine.
Okay.
That is the start codon.
Okay. Thank you, madam.
Okay. Okay, semua dah ada ke? Jude sudah pulang, Jude? You are here?
Oh, yes, madam.
Alright, okay. Um, Okay, so we're going to continue on protein synthesis. Okay, we're going to move on to the second part of protein synthesis, which is translation. Okay, translation. We're going to translate apa yang kita nak translate. We're going to translate genetic code. Okay, translate genetic code carried by mRNA to synthesize protein. Okay, translating the genetic code carried by mRNA to synthesize protein. mRNA ni bawa apa? mRNA ni bawa codon. Codon ni lah yang kita nak translate menjadi protein. Okay, tRNA transfer amino acids. Okay, tRNA transfer amino acids from the cytoplasm to a ribosome. The ribosome at each amino acid carried by tRNA, the growing end of the polypeptide chain. Each tRNA arriving at the ribosome carry a specific amino acid and has specific nucleotide triplet and anticodon at the other. Okay, senang cerita, translation, process to translate genetic code carried by mRNA to synthesize protein. So for this process, we have tRNA. We're going to need tRNA. So this is tRNA. As I said, tRNA at the kaki tRNA, we have anticodon. At the kepala mRNA, we have at the kepala mRNA, at the kepala of tRNA, we have amino acid. Okay. In translation, you need to know about two enzymes. You need to know about the name of two enzymes. So, kita tengok apa dia enzim. Pertama yang awak kena tahu. Okay, the first enzyme that you need to know is amino acid tRNA synthesis. Okay, amino acid tRNA synthesis. This amino acid is function as to catalyze the attachment of amino acid to its tRNA, and this process called as amino acid activation. Okay, before we proceed to translation, every amino acid dia tak ada kepala. Okay, tak lawa, tak nak. Okay, so amino acid activation ini adalah tRNA. Okay. Setiap tRNA dia kena undergo this process, amino acid activation. Okay, so before amino acid activation, rupa dia macam ni. After amino acid activation, rupa dia kita ada macam ni. Dia dah ada kepala. Kepala dia adalah amino acid. Allah, tak nampak. Okay, kepala dia adalah amino acids. That is process of amino acid activation catalyzed by amino acid tRNA synthesis. So you can see that this is tRNA. tRNA will bind to the amino acid tRNA synthesis, and this amino acid tRNA synthesis will catalyze the attachment of amino acid to the tRNA. Okay, dia akan sambung. Amino acid tRNA synthesis ni dia akan sambungkan amino acid. Okay, it will sambung. Okay, it will it will attach the amino acid here to the tRNA. That is amino acid activation. So when this tRNA leave the amino acid tRNA synthesis, we call that as, kita tak panggil lagi dah tRNA, we call this as amino acid tRNA. Kita panggil dia sebagai amino acid tRNA. Okay, that is amino acid activation. Alright. Ribosome. Okay, kita perlukan mRNA, kita perlukan tRNA, and then we have ribosomes. This is ribosome. Ribosome, we have two subunits. Okay, dia ada it has two subunit, large subunits and small subunit. At the small subunit, it has mRNA binding site. At large subunit, it has 3 binding site. It has E site, P site, A site. E is acid site. P is peptidyl tRNA binding site. A site is amino acid tRNA binding site. Okay, tak perlu ingat nama penuh, ingat EPA site. Okay, EPA site. Right? Yang itulah. E site acid adalah di mana tRNA akan keluar melalui E site. P site adalah peptidyl tRNA, maksudnya dia akan pegang peptide, polypeptide chain. A site amino acid tRNA binding site. Maksudnya untuk next amino acid tRNA dia akan masuk yang ni. Okay, yang ni, bilik pertama. Bilik kedua, P site ni adalah kira macam master bedroom. Yang E site ni adalah bilik macam store, bilik belakang. Okay, bilik store macam tu. Okay, so kita tengok apa dia? Ni lah. Okay, tengok gambar ni. E site. E site adalah acid site. So this amino acid tRNA or this tRNA will be discharged from the E site. Okay, dia akan keluar pergi pada E site. This P site, this is where the amino acid tRNA will hold the growing polypeptide chain. And A site, this is where the amino acid tRNA carry the next amino acid. Okay, dia akan pindah. Contoh nanti kita tengok. Okay, during translation, we have 3 steps, 3 stages. First is initiation, elongation, termination. Initiation, permulaan. Elongation, we have 3 sub-stage. Codon recognition, peptide bond formation, translocation. And number 3 is termination. Okay, so kita tengok dulu apa itu initiation. I'm going to explain based on this diagram. Okay, initiation. Okay, what happened during initiation? This is mRNA. Okay, we have small ribosomal subunit, we have initiator tRNA. Kenapa kita panggil initiator tRNA? Sebab tRNA inilah sebagai permulaan untuk translation. Okay, so what happened here is, okay, apa yang berlaku? This mRNA, okay, this small ribosomal subunit, it will bind to mRNA. Then, initiator tRNA will bind to the start codon. Okay, initiator tRNA which bring the anticodon UAC will bind to the start codon AUG. After initiator tRNA bind to the start codon, large ribosomal subunit akan datang. Barulah large ribosomal subunit datang. Bila datang, lengkaplah translation initiation complex. Okay, and this initiator tRNA will occupy P site. Okay, it will occupy P site. For example, tengok yang ni. This is an initiator tRNA. It will bind to the start codon AUG. Okay, it will bind to the start codon AUG. After it bind, barulah large ribosomal subunit datang. Okay, initiator. Initiation. Okay, initiation. Right? Then we have elongation. Okay, elongation. Elongation, we have 3 small steps, which include codon recognition, peptide bond formation, and translocation. Okay, codon recognition, peptide bond formation, translocation. Okay, codon recognition, is when the anticodon or anticodon of an incoming amino acid tRNA base pair with codon of mRNA in the A site. Okay, so what happen is codon recognition maksudnya dia akan recognize the next codon. Contoh the next codon here kita ada UAC. Ini adalah codon seterusnya. Tak nampak. Okay, ini adalah codon seterusnya. So apa yang jadi masa codon recognition, the next tRNA which bring anticodon, kalau sini UAC, anticodon dia akan jadi apa? Sini, okay. Ini adalah codon sini adalah UAC. Apa ni adalah amino acid tRNA. Kalau sini UAC, anticodon kat sini akan jadi apa? Anticodon sini akan jadi AUG. So UAC ni apa dia punya amino acid? Refer balik genetic code table. UAC apa dia punya amino acid? Apa dia punya amino acid untuk UAC? Cari kejap. UAC?
Tyrosine.
Very good. Tyrosine. So, okay. Mana dia? Bukan. Okay. So, kejap. Kita bagi dia jalan dulu. Okay, that is UAC. So, UAC ni dia punya amino acid tadi tyrosine. So, dia akan bawa tyrosine. TYR. So, this amino acid tRNA, it will occupy A site. Okay? Itu maksudnya codon recognition. Dia akan recognize next codon apa dia. So, kita tengok contoh. Tengok contoh yang ni tadi. Sekejap, kita padam dulu. Tengok contoh yang seterusnya. Yang ni GUA. So, dia masuk. Valine. Okay, kita pindah. Masuk. Pindah. Okay, that is codon recognition. Lepas tu kita ada peptide bond formation. This is the second enzyme that you need to know. Peptidyl transferase. Okay, peptidyl transferase. Peptidyl transferase, the formation of peptide bond between amino acid in the A site and the growing polypeptide in the P site. Okay, peptidyl transferase, the formation of peptide bond between amino acid in the A site. Kat sini, amino acid kat antara A site, tak nampak. A site dengan, dekat A site dengan P site. This step remove the polypeptide from the tRNA in the P site and attach it to the amino acid tRNA in the A site. So what happen is, kita tengok. Methionine, valine, ada peptide bond. Then pindah. Valine, ada peptide bond, arginine. Pindah macam tu. Okay. So saya repeat lagi. Ni. Peptide bond. Siapa yang tolong cantumkan? Peptidyl transferase. Then it move. Okay. Move. Macam itulah. Itu peptide bond formation. Then we have translocation. Translocation tu maksudnya dia akan bergerak. Translocate. Pindah lokasi. Okay. During this translocation, the ribosome translocate the tRNA in the A site to the P site. So what happen here? Okay, tengok, perhatikan tRNA in the A site pindah kepada P site. A site pindah kepada P site. Yang tu. Okay. At the same time, tRNA in the P site tadi patutnya dia akan pergi kepada E site. Okay, tRNA will move to E site. Contoh Okay. Peptide bond. Okay. Bond translocate to the P site. Yang ni translocate. Yang patut ni, yang ni tadi yang pergi atas tu, patutnya dia pergi kepada E site. That is translocation. Okay, that is translocation. Tak adalah pula gambar translocation. Nanti saya tunjuk video. Okay, that is translocation. It will move to the E site because it want to exit. Okay, that is translocation. Then we have stage 3, which is termination. Termination adalah proses untuk kita terminate. Kita nak stop, kita nak terminate, kita nak stopkan proses translation ni. Macam mana kita boleh stop? So once it reach stop codon, okay, once it's reach stop codon, for example, kita ada 3 stop codon, uwa, uga, dia sampaikan, dia jumpa salah satu. Katalah dia jumpa uwa, dia jumpa UAG. So when ribosome reach stop codon, the A site, okay, the A site will accept a protein called release factor. Okay, once it reach stop codon, release factor will enter A site. Once release factor enter, this release factor, it will hydrolyze the bond between the tRNA in the P site and the last amino acid of the polypeptide chain. Maksudnya, release factor, it will putuskan, it will hydrolyze this bond. Tak lawa. Okay, release factor, it will hydrolyze this bond. Bond between tRNA in the P site and the last amino acid of the polypeptide chain. Last amino acid yang ni lah. Okay, last amino acid. Okay, the polypeptide, after dia dah hydrolyze, then the polypeptide chain is free. After polypeptide chain is free, the translation complex will dissociate. Okay, small ribosome subunit pergi tempat lain, large ribosome subunit pergi tempat lain, mRNA pergi tempat lain, release factor pergi tempat lain. Okay, that is termination. Okay, contoh yang ni. Okay, sampai je release factor masuk, hydrolyze the bond, disassemble. Okay, I repeat. Once it reaches stop codon, UGA, release factor will enter the A site, hydrolyze the bond, translation complex will be dissociate. Boleh ke guys? Guys?
Boleh.
Boleh aja.
Boleh.
Boleh. Saya pun boleh je. Alright. Okay, yang tu lah. Okay, yang tu. Masuk je pink ni. Pink ni udah buat apa? Pink ni akan dissociate. Alright. Then we have a polyribosome. Okay, polyribosome. Polyribosome, okay, this is a structure of many ribosome attached to single mRNA. Ribosome ni banyak. Ribosome adalah ribosome lah. Poly maksudnya banyak. So dia refer kepada many ribosome attached to single mRNA. Okay, dia satu mRNA saja. Okay, satu mRNA, tapi satu mRNA tu dia ada banyak ribosome. That is polyribosome. Why we have polyribosome? To accelerate the synthesis of many polypeptide of the same kind. Okay, that is the function. Soalan pernah tanya 2 markah. Dululah soalan structure dia pernah tanya 2 markah. What is polyribosome? And state the significance of polyribosome. So the significance, the importance is to accelerate the synthesis of many polypeptide of the same kind. Maksudnya kita akan hasilkan, okay, kita nak percepatkan penghasilan polypeptide chain or protein, okay, yang sama. For example, we want to produce hormone adrenaline. Macam tadi kelas tadi ada bagi tahu contoh. Okay, we want to produce hormone adrenaline. Okay, hormone adrenaline. So what happened, for example lah, okay, for example, want to produce hormone adrenaline. So what happened is we have this polyribosome whereby it will accelerate the synthesis of adrenaline dengan cepat. So kita akan hasilkan polypeptide chain, atau kita akan hasilkan adrenaline dengan cepat bila kita ada polyribosome ni. Okay, after we have polypeptide chain, polypeptide chain tu ataupun protein tu nak pergi mana? Lepas kita ada polypeptide chain, dapat je polypeptide chain, dapat je protein, dia nak pergi mana protein tu? Nak pergi mana? Protein tu nak pergi mana?
Madam, can you repeat again?
Okay, after we have the polypeptide chain, okay, this one, okay, we get the polypeptide. Okay, we have polypeptide chain, we have the protein. What happened? Where is the location? Where is the next location of the protein? Protein tu nak pergi mana?
Golgi apparatus
Why?
to modify the protein.
Betul, pandai Jude. Yes, it will go to Golgi apparatus. Kirtina pun bagi tahu jawapan yang sama. It will go to Golgi apparatus or Golgi body. Why? Because at that organelle, in the Golgi apparatus, it will be modified. Modified packaging, sorting, shipping. So first kita kena nak modify dulu. Polypeptide chain ni bukan kita boleh terus guna. Tak, dia kena pergi modify dulu. Okay, kena alter dulu, kena modify dulu. Nak modify dekat mana? Dekat Golgi apparatus, Golgi body. Then barulah dia boleh guna dalam sel ataupun dia export keluar sel. Soalan pernah tanya sebab tu saya tanya. Okay. Ada soalan lain?
Madam,
Yes.
transfer RNA come from where, madam?
Transfer RNA come from where? Transfer RNA can be found freely in cytoplasm. Dia memang ada dekat cytoplasm. Boleh ke?
Madam, so after they are done with the further modification by Golgi apparatus, if they want to be exported outside of the cell, they use the secretory vesicle and all that, right?
Yeah. The back to endomembrane system.
Okay.
Yeah, yang tu. Transport vesicle, secretory vesicle, yang tu lah.
Secretory vesicle. Okay.
Yeah.
Thank you, madam.
Welcome. Anyone else? Ada soalan? Soalan lagi? Ada soalan tak? Tak ada. Pandai korang. Memang pandai. Saya kagum. Tak ada soalan Tak ada? Okay, kalau tak ada. Okay, sebab saya tak naklah sambung saya tak naklah sambung 6.4. Banyak sangat. Okay, 6.4 banyak sangat. So kalau tak ada, saya tengah fikir. Okay, kalau tak ada. Tak boleh lah nak bagi quizzes pun. Mesti korang kena study dulu. Okay. Tak ada kalau tak ada macam tu kita. Okay, kalau tak ada mungkin kita stop sini lah kot. Boleh?
Boleh, madam.
Okay. Tak adalah stop sini maksud saya. Nanti saya akan assign app puzzle. Okay, satu lagi app puzzle mengenai protein synthesis. Okay, protein synthesis. Lepas tu saya akan assign juga quizzes untuk DNA replication, transcription and translation. Okay, actually 2 quizzes je lah. DNA replication 1, protein synthesis 1. So nanti minggu ni tolong buat. Lepas tu, apa namanya? Lepas tu nanti saya akan post video